So I obviously did my project with Rinky, so that's why he loves me so much. So I'm going to be talking to you basically a bit about Asteraceae, what they are, the daisy family. Do you know that? Oh, they're Asteraceae. So you've got thistles, sunflowers, stuff like that. So we're talking about the plant-pollinator interactions, the structure of Asteraceae, what a floret, what a corolla is, because I don't know if you'll know that already, why we want to study this at all, and what we actually found out. Okay? So, you can see examples of Asteraceae there. It's a massive family. There's over 32,000 species in it. We managed to get data on these three, highlighted by the little stars. So the thistles, can't pronounce that, and I can't pronounce that one either, but we did them. So, yes. So, why is it important to study this? So pollination is massive. You need to learn about pollination to support ecosystems, to support just bees and all the nice butterflies and everything. So we wanted to find out if the corolla length of the Asteraceae species that we studied would influence the insects that visited it. And a little side aim was to see if there was a correlation between the corolla length and the plant subfamily. So whether all the thistles had similar length corollas or whether they were like massively different. And then from research, we did a little literature review, and we found that insect species with longer proboscis tend to visit flower species with longer corollas. So you get longer corollas with longer proboscis, shorter corolla, shorter proboscis. And yeah, that closely related species tend to have similar features. Now, this is one of my favourite photos, one of the little diagrams. So you can see that you have ray florets and disc florets at the top there. There are other florets, so you get filiform, and then you get like bisexual, male, female. We just said any corolla will measure it. And we did ray and disc because they're pretty easy to figure out what they are. So yeah, you've got one line of symmetry with the ray, and then for disc you have 5 lobes which are fused, so it gives you radial symmetry. And then you have different types, so you have radiate heads which have both ray and disc, so that would be sunflowers. Discoid and disciform, which are basically disc florets. And then ligulate, yeah, which is dandelions, if I'm right. Yeah, cool. So this is pretty easy to understand, I think. So you have your ray florets, which are your yellow petals, basically. They're all flowers. Every single tiny little dot is a flower that makes up the massive flower head. So a sunflower is actually hundreds of little flowers, if that makes sense. So you have your disc florets here. You have your corolla, which is about here to here. And you have one on your ray as well. And now this is a lovely little bee demonstrating how it drinks. So you can see that the proboscis has to reach basically to the bottom of the corolla so that it can pull the nectar up. Yeah? Okay. Ooh, we're going again. So in all, I measured about 300, I counted 300 florets from all of these species. So you've got your thistles, dandelion, cornflower, and then at the bottom I just wanted to put these. So these are the insects that visited. Yeah, the kind of main groups that we found. Just nice to visualise it. So Rinky had the pleasure of collecting the visitation data. So I think, how many years did you do it? Like 5 years? More, 5, yeah. He would go out walking and count how many insects on which flowers, where they were at, what they were doing. Well, not really what they were doing, but that's in there. If they were on it, we counted it as they were drinking the nectar. They were interacting with the corolla. So I got one of these years that was 2023, I think. Yeah. And then I analysed that, and we'll get to that. And now for the lab work, this was 18 weeks of maybe 5 hours a week, every Friday, going into the lab, dissecting little tiny frozen flowers that he collected, clearing them, staining them if it needed to. And I wanted to show you how tiny they were. This plate is about that big. So you can imagine how tiny. These are yarrow florets. So you have the disc in the top 2 rows and the ray in the bottom 2, just there. So yeah, it was quite fiddly. So we would clear them if we needed to, which like removes the pigment and lets the light shine through properly. So you can get an accurate measure for like the landmarks and everything that you're measuring. And then if we needed to, we'd stain them. That's spelt wrong, but that's all right. Place them on a slide, cover slip them, and then we'd use this fancy machine. So it's a normal light dissecting microscope, but it's got an iPad attached to it, which is very fancy, and he had to fight for it. People were very jealous. So to start off with, we measured 2 lengths. So we measured L, which is the length of the disc floret, and then we measured L on the ray floret, which is this bit, because this is the part that basically the insect needs a proboscis long enough to reach the bottom of here to get to the nectar, because it's produced in nectary cells down here and here. But as we went along, I found that thistles, particularly, would have disc floret shaped like this. So you'd have a really, really narrow corolla, but you couldn't really fit anything in. So it's kind of useless to measure all of this. So I measured this as well, L1, which I call the effective length. Because this is basically what I thought an insect would be using. They wouldn't go all the way down here because you physically couldn't, because you've got the anthers and the style and the stigma in there. So it was basically useless. But we didn't end up using that length because it was a tiny study. We only had maybe 3 species that were shaped like that. So we used the normal L. But I just thought I'd put that in, it was kind of fun. So these are some slides. So you have yarrow at the left and the right, and then you have some kind of thistle in the middle. So can you see what I mean about it narrowing in the middle? Yeah? So, yeah. Lovely. Now, this is the lab data. It's a lot. But if you look at it, you can see it's separated into CA, which are your thistles, mostly. True thistles only have disc florets. So some of them are kind of weird ones. Then you have your tansy, your oxeye, then you've got your dandelion and nipplewort, which are their own separate sub-family. So you can see that there's clearly differences in how long the corollas were. These are all measurements in millimetres. So it's tiny, tiny, but there is still differences if you're looking at that level. Now, this is my favourite graph. I'm very proud of it. So this summarises the visitation data that we got, and I've ordered it. So the top with your spear thistle, this is your longest corolla, and you get shorter and shorter, dandelion, ragwort, Michaelmas, and yarrow's your shortest corollas. And you can see with the purple, you can see kind of 3 main groups if you're looking at what makes up them, because you've got mostly bumblebees visiting the top 3, which are your thistles. I'll explain the legend first. BB means bumblebee. So these are all the different species that we had visiting. And in the first group, your purple, your thistles, your longer corollas, that was mostly bumblebees. You had the odd fly, hoverfly, of course you would, because they're over everything. Then your second group is your yellow, So that's your dandelion, ragwort, tansy and yarrow. These ones, you can see really high percentages of hoverflying flies. Now these ones are like short corollas, but they're also yellow and white flowers. With your thistles, they're all purple and blue. So you'd be getting more bumblebees anyway. But we don't know if it's just because of the corolla, or if it's the colour, or if it's both. It's probably both. But it's a tiny study, so we couldn't really say for definite. And then you have an outlier, a little random group, your Michaelmas daisy, that honeybees absolutely loved. They went for it. Now, I thought I'd include the photos just to prove. Obviously, these ones have the longer corollas. They're your thistle family. Then you have your hoverfly-loving ones, your tansy, dandelion, and then your Michaelmas, which honeybees loved. So it was a tiny study, but we still were able to find good trends. So it's kind of indicative that bumblebees like a longer corolla. And yeah, thank you for listening.